In this video, we're looking at blood vessels. So our learning objective to describe the layers of the arteries and veins and describe the difference between arteries, arterioles, and metarterioles. Describe where most of your blood is stored. And we also want to describe what goes on at the arterial and venous ends of a capillary. Answer these questions. If 5 liters of blood moves to the arterial end of capillaries in 1 minute, how many liters of blood is returned to the venous end? And describe 3 types of capillaries. Okay, so let's go through that. And so when we look at the blood vessels themselves, there are your arteries and there are your veins. Remember by definition that arteries carry blood away from the heart and veins return blood to the heart. And so the thing about this is that each of these blood vessels has 3 layers, and they're the same 3 layers. On the innermost layer, the part where really the blood is touching, we call that the tunica intima. Some books will call it the tunica interna. It's the same thing. And the next layer is the tunica media, and then the outermost layer is the tunica externa. And you can see that, again, arteries as well as veins have those 3 layers. What you want to know is within the tunica intima, the endothelium is going to make up that innermost layer. And again, this is the part that directly faces the lumen of the blood vessel. That's the endothelium. The endothelium is continuous with the endocardium of the heart. Underneath that endothelium is just another layer underneath. We just call the subendothelial layer. And as part of that, there's some elastic tissue there to give a little bit of elasticity to these vessels. Tunica media is the next layer, and that is made up of muscle. Now, what kind of muscle is it made up of? You know that there are 3 muscle types: cardiac muscle, skeletal muscle, and smooth muscle. These are made up of smooth muscle. Tunica media is made up of smooth muscle in both arteries and veins. And then the last one is the tunica externa. So right over here, we've got that elastic lamina, some more elastic tissue between tunica media and tunica externa. And tunica externa really is just a bunch of connective tissue that wraps up the whole darn thing. So from inside to out, tunica intima or tunica interna, then tunica media, followed by the tunica externa. So what we'll see then is that transition between arteries and veins. So what we're going to talk about, let's for the rest of this video, let's just from here on out ignore the pulmonary circuit. Okay, so if we ignore the pulmonary circuit, it's safe to say that all these arteries carry oxygenated blood and all these veins then carry deoxygenated blood. So what you want to imagine is good oxygen-rich blood coming through here, eventually making its way into this network of blood vessels, which we call capillary. And notice what happens here. This artist depicts this bright red color going into a blue color. And really what this is representing the loss of oxygen from these blood vessels. So what do we mean by loss of oxygen? It means oxygen is leaving these blood vessels to go out into the surrounding tissue, the interstitium. So from there, deoxygenated blood then is going to enter into the veins, which then will go back up toward the heart. So these are the layers of the arteries and the veins. There's different types of blood vessel. So here there's with respect to arteries and veins, we have these elastic arteries and muscular arteries, and we'll see that the makeup is a little bit different. If you look at this relative tissue makeup of endothelial tissue versus elastic tissue versus smooth muscle versus that kind of fibrous connective tissue, the overall kind of distribution of tissues, I suppose, within these blood vessel walls is a little bit different. And I don't need you to memorize that. I don't need you to know the difference between elastic arteries and muscular arteries. But what I want you to see are arterioles right over here, as well as capillaries. Arterioles and then capillaries right over here. So when you look at arterioles, what I want you to see is that there's a ton of smooth muscle in arterioles. And what we'll find is that once you mess with arterioles, this is going to be the main determinant of whether or not a tissue gets a whole bunch of blood or not. That's what we're really going to consider. Capillaries themselves are made up of only endothelial tissue. So capillaries have only endothelial tissue within them. If you look it through, look, there's no elastic tissue, there's no smooth muscle, there's no connective tissue. And why is that? Because capillaries, that's where exchange happens, meaning that this is where the good stuff is going to leave and then the waste product is going to enter in. And so stuff leaves and enters in at the capillaries. You don't see any gas exchange occur at arteries and such because these walls are just way too thick for something to get out. But the capillary walls are thin enough that through the capillary, that's where things can exit and enter. Through venules and veins, again, there's no exchange happening because the walls are just too thick. And so the system that we see here, if we lay it out, we've got the heart, and then we have the blood going into arteries, eventually going into arterioles, eventually going into these things called metarterioles. Now, what are metarterioles? Metarterioles themselves are the pipe that immediately comes before capillaries. So we have capillaries and then venules, postcapillary venules, back to veins, back into the vena cava, which goes back to the heart. So this is kind of the overall layout of the vascular system. Arteries into arterioles, into medarterioles, into capillaries, into venules, then into veins, which goes back to vena cava, back to the heart. So when we look at distribution of blood at any given point in time, if you were to take a snapshot, then this is showing you the snapshot of where your blood is right now in a snapshot. So most of your blood, 60% here, is contained within systemic veins and venules. So most of your blood here is in the venous system. So what I will tell you, we've talked in previous lectures about this idea of venous return, venous meaning veins here. And so I gave you examples of things like Bainbridge reflex and Starling reflex that are kicked in as a consequence of increased venous return. And all that means is, let's say this part right over here, let's just indicate that to mean venous return. That's a weird arrow, but let's say back into the heart. Well, in some situations, let's just say you have, whatever, 5 liters per whatever unit of time that returns back to the heart. But under other conditions, instead of 5 liters, you're now going to have, let's say, 15 liters. And again, I'm just making up these numbers, so don't memorize these numbers. My point is you can increase the amount of venous return. How do we do that? Honestly, it's pretty simple. All we do is vasoconstrict the veins. If we vasoconstrict the veins, we just leave a lot less room in there, meaning that blood needs to continue to go forward, and that can then be distributed into other areas of the body. So at rest, though, I do want you to know that most of your blood is contained within systemic veins. Now, what I want to do is backtrack just a little bit and talk about this thing called medarterioles. So let's look at this. What we have here is a arteriole, and it says terminal arteriole here, meaning there's different types of arterioles, which we don't care about. We're just going to say arterioles. This pipe right in between, right over here, is called a metarteriole, and then everything else here, all these little vessels, are the capillaries. So what is the medarteriole? The medarteriole is almost, you can think of it as like maybe in some ways like the main neighborhood road that goes through the neighborhood. If these are the neighborhoods up and below, then this is like a main neighborhood road. And so this so-called thoroughfare channel. And so the medarteriole carries blood through here and allows blood to then enter into the capillaries. Now the thing about it is that you got sphincters all over the place. Sphincters are just little kind of circular bands of smooth muscle, and these precapillary sphincters, if they clamp down shut, if these muscles contract, then you see that blood cannot go into the capillaries and it just passes through the medarteriole back into the venous system. On the other hand, if the precapillary sphincters are open, then now I can perfuse tissues. Again, remember the word perfusion means send the blood supply to tissues. And so that's what we've done here. Here, precapillary sphincters are open. Here, precapillary sphincters are actually closed, and so I don't get perfusion of this tissue. Now, why might I do that? The reason I do that is because ultimately, look, you've got 5 liters of blood to spread throughout your body, and it's just not enough blood to perfuse all tissues maximally. Just like, look, there may be a lot of things that you want to do in your life, but you have a limited budget. So you allocate some of your budget to this activity, some of your budget to rent, some of your budget to this. And so you don't get to spend as much rent money as you want, as much food money as you want, as much entertainment money as you want. It would be great, but that's not the reality for most humans who live on a budget of some kind. Similarly, your system has a budget with respect to blood, and so you can't send a whole bunch of blood to all tissues all at once. It just doesn't work that way. So in that situation, let's say in a fight or flight response, let's pretend this is your stomach, let's say. And look, if I'm running away from a bear in a fight or flight response, I don't need to spend a lot of time thinking about food digestion, right? That I'll leave for your rest and digest response. And so therefore, what do? I close down these precapillary sphincters. I'm also going to vasoconstrict upstream. And when that happens, I don't have a whole bunch of blood sitting here in my the interstitium of my stomach and intestines and things like that. That way I can reallocate them to places that matter in a fight or flight response, like liver and skeletal muscles. All right, so let's see. Where we're at so far is that we've described the layers of arteries and veins. We talked about a little bit of the difference between arteries, arterioles, and medarterioles. We said most of your blood is stored in veins. And what I'm going to do is I actually need to talk about this thing over here, and I'm going to save these 2 learning objectives for a next video. Okay, so let's describe the 3 types of capillaries. The 3 types of capillaries are listed right over here, and you don't have labels on them, so let me go ahead and label them right now. And now that we got those labels down, here are the three types of capillaries that we see. So we have your kind of traditional, standard, run-of-the-mill capillaries, and these are your continuous capillaries. What I want you to see is that here are my endothelial cells. And remember I told you before that these capillaries can be so small that red blood cells pretty much go through single file because they're just not that big. And so here is an endothelial cell. These are other endothelial cells. And they sit really, really close to one another. We have some other cells over here, these pericytes, which we'll see again in the renal system. And these can basically regulate the level of permeability of these capillaries. So anyway, these are continuous capillaries, and those are probably the most famous ones. Fenestrated capillaries, what we're looking at here, again, we've got pericytes on all of these things, but fenestrated capillaries, if you look at it, it looks like, whoa, it's weird. There's a bunch of holes here. And in fact, there are holes. So these holes have a fancy name, and they're called fenestrations, which is why they're called fenestrated capillaries. And so when you see something like this, what I want you to imagine then is, well, which one is leakier? And which one of these is permeability higher? And it's probably no surprise to you that the one with holes in them are more permeable. So fenestrated capillaries have increased permeability compared to continuous capillaries. The last type that we see are sinusoidal capillaries. Sometimes they're just called sinusoids. And what we see here are actually, again, we see fenestrations, but this time they're huge. Instead of being these tiny pores, they're huge. So here we probably shouldn't call them fenestrations, because these are actually the fenestrations. But the point is there's a quite big gaps within the cells themselves, as well as gaps between cells. When we see something like that, then these are the leakiest capillaries of all. And there's going to be time and place that we want to see that. So really all I want you to see here when we look at the three different types of capillaries, this last learning objective, what I want you to see then is that I've got continuous, fenestrated, and sinusoidal capillaries, and they're all in different locations of the body, but kind of your standard run-of-the-mill capillary are going to be these continuous ones. These are more in special circumstances, and the difference between them is the degree of permeability. And that should wrap up this video. We'll go ahead and make another one that goes into just these two learning objectives right over here.